Living cells are like miniature cities with constant traffic along tracks connecting different parts. Vehicles inside cells are molecular motors proteins that transport cargo moving on tracks. One of them is the microtubule. Microtubules have a distinct plus and minus ends. Kinesins, a type of motor, move typically to the plus end. Kinesin-1, a widely studied kinesin, has a linear structure with a motor head that walks, an intervening domain, and a tail that binds cargo and microtubules and regulates the motor. We wanted to understand how the tail affects transport when multiple motors are acting simultaneously. We used a technique, the gliding assay, where motors are fixed on a glass surface and microtubules move on them when ATP is added. Just like a microscopic escalator, the technique's name originates in the apparent gliding of microtubules when observed under a microscope. When we attached Kinesin-1 such that its head or tail was equally likely to point upwards and contact microtubules, we observed microtubules moving in a stop-and-go manner, occasionally bending, looping, and even oscillating like a wave. We hypothesized that kinesin was behaving like a coin toss where a head or tail are equally likely. We predicted multiple kinesin heads and tails binding simultaneously to a microtubule would result in a tug-of-war and cause bending. We tested this by varying the proportion of proteins that had either only the tail or only the head domain in varying proportion. All tails resulted in static microtubules, while increasing the proportion of heads increased velocity. All heads showed unobstructed transport. Interestingly, a 50-50 ratio resulted in stop-and-go motion, bending, and oscillations as seen with Kinesin-1. Thus, we find random statistics and the mechanics of motors and microtubules together determine how motors act collectively. In future, we hope to examine the effect of this inside cells.